Hello everyone, my name is Maria José Silveira and I'm glad to present the work of my PhD that I have been developing during the last 2 years, which is related with development and characterization of CEA-targeted nanoparticles as a novel chemotherapy for metastatic colorectal cancer treatment. And I cannot... Oops. And colorectal cancer is the second deadliest cancer in the world, and specifically in Portugal was the most common cancer in 2020. And as also known, colorectal cancer arises from a premalignant lesion in the mucosa of rectum or colon, and through the acquisition of genetic alterations in the oncogenes or tumor suppressor genes, such the loss of APC gene, this neoplasia arises. Moreover, the immunosuppressive tumor microenvironment surrounding this tumor is also important due to the impact that it has in the progression of the disease and also in the metastasization process. So we can correlate with the high mortality of this cancer with the metastasis events, with the appearance of metastasis. And regarding to the therapeutic approaches, such as target therapies and emerging immunotherapies, chemotherapy is still one of the most therapeutic approaches used, and chemotherapeutic regimes based on 5-FU regimes are highly used, however, with a great toxicity, which leads to a limited bioavailability and low tumor-specific selectivity, elucidating the needs to exploit alternatives, and this is where nanomedicine came in. And regarding to the nanomedicine, nanoscale delivery, we will focus on nanoparticles and specifically in polymeric nanoparticles. And PLGA nanoparticles could provide a drug formulation in a polymeric core, biodegradable and biocompatible. And when shielded with a PEG, a polymeric pad, it will allow the immune system diminishing the immune recognition and also its degradation of the nanoparticles, allowing also the surface modification of the nanoparticles with the ligands. So our strategy aims to target colorectal cancer cells using polymeric nanoparticles to deliver the 5-FU chemotherapeutic drug. Therefore, we intend to encapsulate 5-FU into the PLGA core, which will be then shielded with a PEG, which will allow the attachment of a specific fragment antibody towards the CEA expressing colorectal cancer cells, which is actually an overexpressed glycoprotein present in the most colorectal cancer patients. And for this, we perform the chemical conjugation of the polymer with the anti-CEA single-chain fragment variable, which was provided us by our collaborators, Kerry Chester teams. And then we performed the nanoparticles production and the 5-FU encapsulation by double emulsion technique. And regarding the single-chain fragment variable, we are using I would like to emphasize or highlight the expertise of our collaborators in the antibody engineering, and I would like to explain a little bit of this strategy. The single chain fragment variable was produced by phage display technology and also by recombinant antibody technology. They used a heavy chain variable domain and a light chain variable domain which were then linked by a flexible polypeptide linker. And after that, they characterized this single chain fragment variable, and they achieved a fragment with a high stability and with a low size, with around 57 kilodaltons. So, due to this small size of the fragment, this type of fragments are considered one more effective strategy to target solid tumors due to the easily internalization and penetration through this type of tumors. So, in order to perform our accurate these hypotheses, our collaborators performed a covalent conjugation using a pegylated spions through the sodium periodate method. And after this functionalization, the nanoparticles were characterized, and the specificity targeted to the CEA positive cancer cells was also achieved. So going back to our strategy, I would like to start to explain a little bit of our chemical structure of our CEA target polymer. So I would like to start to refer that our PLGA PEG was used with a malamate group for the TOL Michael addiction to conjugate with the single chain fragment variable with the PEG. So after this, we performed the NMR evaluation of the polymer, and we achieved peaks from the single chain, specific from the single chain fragment variable in our final spectrum. Moreover, we performed an evaluation through the HPLC, and we achieved a conjugation efficiency with around 80%. After this, after the synthesis of the polymeric conjugate, we proceed to the production of the nanoparticles using an optimized double emulsion technique, and we achieved 5FU loaded PLGA single chain nanoparticles with around 170 nanometers and with around 0.2 polydispersity index and with around 5% of drug loading. Moreover, we characterize our nanoparticles as according to its morphology, and we achieved and we saw that our nanoparticles were also spherical as intended. As a conclusion, we have synthesized a biological target polymeric conjugated with high conjugation efficiency suitable to manufacture a colorectal cancer nanomedicine. 5FU loaded nanoparticles were produced by double emulsion technique with a satisfactory drug loading and association efficiency of the drug, and with around 170 nanometers, 0.2 polydispersity index, and also with a spherical morphology. Moreover, as a future work, we'll intend to perform release and cytotoxic assays. And also, we would like to highlight that we also performed a complete screening of CEA-expressing colorectal cancer cells for further evaluation of the specificity of the ligand and our nanoparticles. As a final remarks, I would like to share with you the overall goal of our project, which is related with the development of combinatory strategy using the developed CEA colorectal cancer cells.
You know, part of my science and engineering have been, I think, an intimate part of my life since I first decided that I wanted to be a science or an engineer.
Okay. Can you see my screen?
Yes. Yes.
So, and also, we also would like to associate or develop an immunomodulatory strategy using nanoparticles, immunomodulatory nanoparticles, to act in the immunosuppressive tumor microenvironment, reverting the immunosuppression signals, recruiting T cells, and helping to kill the cancer cells. So finally, I would like to acknowledge this work mainly to my supervisor, Professor Bruno Sarmento, and also to Professor Maria José Silveira. I would also like to thank you, Professor Carrie Chester, to kindly send us the single chain fragment variable, all the other people involved in the work, our research group and funding, and also to you for your attention and for the opportunity to share and present my work.
Thank you very much. Questions, please.
Okay.
To raise your hand or to just to type your question in the chat.
Maria José, I have one question.
Ivana, please.
Yeah, did you check or did you compare the efficacy and toxicity of 5-fluorouracil with the nanoparticles loaded with 5-fluorouracil?
No, we performed the characterization of our nanoparticles with the target nanoparticles, and we had a lot of optimizations. So the next work that we are just starting is related with toxicity and the evaluation of the toxicity of free drug and the encapsulated drug into our nanoparticles and our functionalized and non-functionalized nanoparticles. So,
and before this testing, did you already perform the stability testing of how stable is the attachment of 5-fluorouracil to the nano surface?
No, we didn't perform it yet.
Okay, so because this would be highly important to see the stability of in different media, this attached drug.
And we also need to evaluate the stability of our single chain in the nanoparticles, and we are just performing it. So we have a lot of things to do.
Thank you. Good luck.
Thank you.
Thank you very much. Any more questions? Please pay attention to the question asked in the chat. The cytotoxicity of 5-fluorouracil is very low, and you need very high amounts of it to be effective. Why did you choose this drug? Did you plan to incorporate another synergetic drug, maybe?
Sorry.
You can follow it in the chat.
I'm trying to check the chat, but I can't.
The main
so rate is very low, and you need very high amount to be effective. Why did you choose this drug? We chose this drug because it's one of the most drug used to treat not only colorectal cancer but also to treat a lot of cancers. And we know for the clinic and also for the research that are described that this drug has a lot of toxicity and a low bioavailability and is associated also with a lot of resistance. So we would like to encapsulate not only to decrease the toxicity of the drug but also to decrease the resistance, and also we know that 5-FU in a free drug can increase the PD-L1, for instance, expression in the cancer cells, and maybe with this encapsulation we can decrease this PD-L1's formation and also the resistance of the cancer cells to the treatment. So as it could be the most used, there are a lot of benefits to encapsulate it, in our opinion.
Any more questions? Okay, so if there is no more questions, thank you very much for your presentation.